could show how much it liked social behavior by having these two little holes, and it could kind of poke a hole, and if it did, it would open a little door. He's going to poke right there, and the door goes up, and he gets to sniff his little friend for 10 seconds. So it's a way of sort of quantifying how social the mice were. And generally, they were fairly social, so that was pretty normal in the mice. Maybe there's some subtle differences in the males. We also wanted to look at learning, and we looked at things. There are a lot of classic tasks in mice to look at how well do they remember their locations, how well do they remember a shock, things like that. And the mice seemed pretty normal on their memory for locations, but it wasn't very sensitive to learning. So we tried to develop something that was a little bit more challenging for the mice. And so my friend Lex Kravitz invented this little device, a little toy that goes in their home cage where they can learn to play a game. In this game, they poke their nose in a hole here, and they'll get a little prize. So here's a little mouse doing this. Oh, ça va marcher, non? Well, this video is not working, but what it would show is he pokes his nose in the hole, and a little piece of food drops out, and he excitedly goes to eat it. We can make the game a little harder by having two holes. And we make it so that initially the mouse learns that the prize is on the left. And he'll push and he'll push and he'll push and get more and more prizes. And then we will trick the mouse and move it to the right. And then after about 30 tries, we'll trick him again and move it back to the left. And so they have to learn to go back and forth and be flexible and be able to change their behavior as the demands change. And the thought was maybe with MIT1L, they have more challenges in changing their behaviors. So we change kind of every 30 pokes. Oop, the little mouse moves. So this is what the siblings look like, the brothers and sisters. So here's which side is getting the reward. It's either getting it on the right or the left or the right or the left. That's the black line. And here's the behavior of the mouse. They initially don't know what to do. They know they get some rewards. So they try both sides. But over the course of days, they learn, I should switch to the left. I should switch to the right. I should switch to the left. The mice with the MIT1L mutations had a lot more challenges with this. They did eventually learn it, but not ever quite as effectively. They tended to kind of stay a little more in one place and have a little bit of trouble switching back and forth. So we think this is kind of a sensitive measure of that learning ability and repetitive behaviors. We also did some brain imaging, and as reported in some of the patients, there's sometimes a tendency for slightly smaller brains, but grossly these things are pretty normal. They're not missing major structures. It's pretty subtle. So it's mostly building a brain you would expect, just a little bit smaller. And we're starting to try and do this at higher resolution now and actually measure the counts of different kinds of cells in the brain to see if if all the normal cell populations are using these new spatial transcriptomic techniques that I might have to tell you about in 2 or 3 years because we're just starting these experiments. I guess one last, so we were doing all this to be able to understand if the mice have the same challenges, because then it gives us a platform to test lots of therapeutics. There's only one Jake, so Dr. Constantino can only try so many things with Jake, but having the mice, this gives us the opportunity to try lots of different things quickly and see what might work. And so seeing that they have similar challenges then gives us we can repeat those same assays with different treatments, as Dr. Mall showed you in his experiments. So we're hoping that'll be something we can do. The other thing that's nice about mice is they have much shorter lifespans, so we're able to look at kind of longitudinally how does this affect development. And I think there was some good news there. I mean, to the extent that mice are similar to humans, we see a very similar lifespan in both them and their siblings. It's not very different. So because sometimes some neurodevelopmental disorders also come with heart conditions or things that affect later in life. We didn't see any of that here, so that was reassuring. So they have many of the same challenges. Why? Well, we think the smaller brains may be because of a little decision during brain development. There are cells who proliferate and double to make more cells to grow the brain when it's young, and there's other cells that differentiate to make neurons. If the cells switch too early to making neurons, you don't quite grow the brain as much as you would like to, and so we think that might be why the brains are slightly smaller, is an early switch to making neurons. What does the gene normally do during development? MIT1L is a gene who controls the expression of other genes. It can be both a gene turn on some genes, and it can also turn off genes. And so that's a little confusing because going in, we thought it would be one or the other. It turns out it's both. It does, at some genes it turns them on and some it turns them off. So there will still be a lot of work to do there to understand what its targets are and why it plays this strange role. Generally, I think both labs that study this saw that it turned on and off very similar genes, which are genes involved in the maturation of neurons. And I think a broad thing that we're seeing is that just like the neurons are born too early, they kind of rush into being neurons, but then they don't really mature all the way as you track them over time. So it seems like they don't quite get to the mature state in the brain from what we can tell in terms of their gene expression. Chris Kroll, our colleague, saw similar things in cell culture. So we think this is true also in the human neurons, as best as we can tell, in terms of staining with markers of maturation, for example, like this kind of MAP2 staining. So that's where we've gotten so far in the last 5 years. I think it's kind of been a lot of platform building to try and be able to create an opportunity to test different treatments with these different platforms. We're looking forward to working with Dr. Mall in our next round, where he in parallel made a different patient mutation. And so we have basically 2 mutations to study. We're hoping to use those in parallel. And the goal for our next 5 years, if funding for science in the US does not collapse, which is at a fair amount of risk at the moment, is to try and understand a lot more of the missense variants. Those variants where we don't know whether or not they have exactly the same consequence as someone who's lost MIT1L levels. The other thing we want to try and do is use what we've learned about MIT1L gene expression to build better diagnostic tools for those missense variants. Looking in the patient records, it seems about half of the variants are missense. And for those, we don't really know if it's exactly the same syndrome or if it's doing something different. The missense variants, for example, might only activate genes or only turn off genes instead of doing both things. And so that's something we want to study in depth so we can help people get better diagnoses and figure out whether these are similar related disorders or very distinct. And finally, take these platforms we've built and try and screen for medicines that'll address these challenges. We have some high risk, high reward studies also about trying to test variations on gene therapies with RNA therapeutics. Because it seems like if you're making too little of the protein, one thing you might want to try is just driving the creation of more of the protein, simply adding it back. But it's unclear the right time to do that. If MIT1L is mostly needed in early development and you add it back later, maybe it doesn't help so much. So the mouse will let us to tease that out if we can build the right tools to let us turn the genes on and off. And that's an area of active research, as well as trying to develop RNA-based therapeutics that would be translatable from mouse to human. And I have a variety of students in the lab who've gotten very excited about this. I think one of the best things about this was a turn from my lab. I was a fundamental, basic researcher in kind of gene expression in the brain, but working with Jake and Dr. Constantino gave me a chance to still explore that fascinating biology, but also do something that had an impact on patients and connections with families. And I think that meant a lot to us also. We had the first group of families come to St. Louis in the US, some of them who I see here today. And I think that was really enlightening for us and changed how we think about the research we do. So we learned a lot from you guys who are really the experts in the disorder. And I think it got the students very excited. So we now have 4 different people in the lab working on MIT1L. We have a physician scientist who's joined us, who's going to talk next and tell us about that. So we're very excited about our continued studies going forward. So I want to thank my team. I've told you about Chris Kroll and Susan. I think the people who really led the work on the former population of students who led the work were Alan Yen, Jiayang Chen, and Simona Serafinoska, and of course the group that are working now on it, like Allison and Colin and Mary. And a new student just joined who's all excited to work on MIT1L. So I want to thank the lab and thank you for your attention.